Olá, boa noite a todos. Muito obrigado pela presença. Eu sou o William Hideki, gerente de produtos do Valeda, na Adapt. Estou junto aqui com o Dwayne Morrison, que é o vice-presidente de vendas da Lumithera. Estamos aqui com os doutores. Sejam muito bem-vindos, né, a este webinar, onde teremos a professora Janis Eells, da Universidade de Wisconsin, Milwaukee, e o doutor, ou PhD em farmacologia, Dr. Clark Tedford, presidente e CEO da Lumithera. Vão estar apresentando os dados e todos os parâmetros que foram levantados nessa última semana sobre o estudo de 24 meses do LIGHTSITE III que foi submetido ao FDA. Mais uma vez, sejam todos muito bem-vindos. Muito obrigado pelo tempo de vocês, por estarem aqui conosco essa noite. E deixo a palavra agora com a doutora Janis Eells, professora da Universidade de Wisconsin, Milwaukee, em farmacologia e formada em farmacologia e fez o PhD em farmacologia pela Universidade de Iowa. Bom, muito obrigado a todos. Vamos ter toda uma boa sessão. Todas as perguntas e respostas podem ser feitas através do Q&A que aparece aí para vocês. Podem fazer as perguntas em português ou em inglês. A vantagem de fazer em inglês, eles mesmos vão acabar lendo. Mas se preferirem escrever a pergunta em português, fiquem à vontade. Eu vou fazer a tradução dessas perguntas ao final da sessão para os apresentadores. Muito obrigado. Uh, Professor Diniz, the audience is all yours.
All right. Clark, can you move the slides ahead, please?
Yes, I will.
Okay, let me start here. So I want to start with a description of photobiomodulation and then give you a review of the preclinical work that our lab's been actively involved with for about 25 years. So photobiomodulation, it's an established and a non-invasive light energy-based technology that has thousands of published articles detailing cellular mechanisms and effects in a variety of medical conditions. We're going to focus on those relating to the eye. Next slide, please. There's been a global explosion of research in photobiomodulation in the area of retinal disease between 2000 and 2024. Over 1,000 published articles on photobiomodulation itself, and over 200 of those have been on photobiomodulation in ophthalmology. And I'd like to point out that these have been published in top tier peer-reviewed journals, Journal of Biological Chemistry, Retina, Ophthalmology, Proceedings of the National Academy of Sciences, to name a few. They include studies of experimental models of retinitis pigmentosa, bright light-induced retinal injury, which is a model of AMD, retinopathy of prematurity, retinal aging, age-related macular degeneration, and diabetic retinopathy. Next slide, please. We also have, I'd like to sort of tout our recently accepted paper in Frontiers of Ophthalmology. It's a special issue on diagnostic and therapeutic applications of visible and near-infrared light for the retina. And we have a review paper that focuses on the use of photobiomodulation in ophthalmology that gives an overview of basically the bench to bedside research, the translational research, reporting in detail on dozens of clinical reports, looking at the beneficial effects of photobiomodulation in ocular conditions, including a review of the preclinical work that focused on the mechanistic effects, a review of preclinical evaluations in animal models of ocular disease, 27 of the peer-reviewed articles detailing PBM photobiomodulation in ophthalmic conditions, including studies, 11 studies in AMD, 10 of these studies are randomized controlled trials. So evaluations of age-related macular degeneration, diabetic macular edema, myopia, amblyopia, retinopathy of prematurity, retinitis pigmentosa, Stargardt's disease, central serous chorioretinopathy. So you can see that the research and the application of photobiomodulation is expanding. Next slide, please. So let me take you sort of back to the nuts and bolts here. How does this work? What is photobiomodulation? The term, if you divide the term up, photo, light, bio, life, modulation, change. And that is, the reason that we selected this term rather than low-level light therapy or low-level laser therapy is that it really encompasses the entire field. And it's the process by which specific wavelengths of light are absorbed by cellular photoacceptor molecules, and that results in the activation of signaling pathways that basically culminate then in changes within the cell. Now, how this has to happen is you have to sort of have two steps. First, these photons, the light has to be, the light waves or particles, it's both, have to be absorbed by a photoacceptor molecule or molecules. And then it has to change or modify the function of that. It has to alter that molecule and set off a chain of responses. Now, photobiomodulation is all over the natural world, essentially. You think of photosynthesis in plants, light interacts with the photoacceptors in the chlorophylls, in the chloroplast, in the plant cell, and causes cell growth. And essentially, chloroplasts and mitochondria, where the light acts in terms of far red, near infrared light, are really evolutionary kissing cousins. So it shouldn't be surprising that light would not only affect plants, but would also affect us. Many of you in the audience are ophthalmologists, so you know that phototransduction occurs in vision. So there is photobiomodulation in vision. A single photon of light can interact with the rhodopsin molecule or the conopsin molecule, turning on the transducin system and working through a G protein mediated signaling system, basically open a cyclic AMP gated ion channel, and what you see is a hyperpolarization which sets off with the photoreceptor the entire cascade that results in vision. What we're going to focus on is far red and near infrared light, primarily yellow also, acting on mitochondria at the level of the electron transport chain in the mitochondrion, specifically at the level of the enzyme cytochrome C oxidase, which I'll tell you more about, and setting off a signaling cascade. So there are two effects that we focused on in my laboratory and other laboratories. One is, I always think of it as you get two things for the price of one here. You turn on mitochondrial function by stimulating cytochrome oxidase, and you alter the redox status of that, and you make the enzyme, the entire electron transport chain, more functional. So you increase ATP synthesis, you increase the mitochondrial membrane potential, which drives ATP synthesis, and you get an immediate acute effect on making that mitochondria happier and more productive. Then you get the long-term effect. At the same time that this is happening, there's the release of either superoxide, nitric oxide, or possibly both that activates transcription factors. Those transcription factors then go to where transcription factors go, the nucleus, and they bind to the DNA, they alter gene transcription, and they alter protein synthesis. And essentially, they create a stable microenvironment. A colleague of mine used to say, all of the good proteins get turned on, all of the bad proteins seem to get turned off. The cell is responding to this stimulus by protecting itself, and there's improved cell survival. Next slide, please. So as I just said, mitochondria are the primary target of photobiomodulation, and what you can see on the right-hand side of the slide is a depiction of the cytochrome C oxidase complex. It's called complex 4. It's the terminal complex. It's the major intracellular photoacceptor for photosignals for yellow, red, where it's mostly we focused on red and near infrared light. It's the terminal electron acceptor in the respiratory chain. And so it's, the electrons come bouncing through the electron transport chain. They basically generate an electrochemical gradient that drives the synthesis of ATP. And the final electron acceptor is cytochrome oxidase. It catalyzes the transfer of electrons from cytochrome C, you can see in the green, to molecular oxygen. And that's that last step. And then ATP synthase makes ATP, and that's the energy currency for the cell that drives all the reactions in your body, basically. The absorption of photons by this cytochrome oxidase increases, starts this whole cascade. And you see reactive oxygen species generation. That would be a superoxide molecule. You also see a photodissociation. It knocks the nitric oxide molecule from its binding site, resulting in, because the nitric oxide molecule is kind of the modulator of how fast the electron transport chain runs. And so if you bounce it off, you get an enhancement of activity. The enhancement of activity means you make more ATP, means you've got more energy. The other thing is it gets more complicated. Cytochrome oxidase doesn't just have one activity in terms of generating water here, in that it also can function as a molecular switch and essentially synthesize nitric oxide in response to low levels of oxygen. So I'll tell you a little bit about that too. So it's a complicated molecule. It's people have spent their entire academic careers just studying this particular part of the electron transport chain, because it seems to be the mother controller of the electron transport chain. If cytochrome oxidase isn't happy, it's kind of like if mama isn't happy, nobody's happy. If CCO isn't happy, the rest of the electron transport chain isn't happy. Next slide, please. So we know that cytochrome oxidase, one of the strongest information, or I think the strongest support, there's a lot of support for the idea that far red and near infrared light interact with cytochrome oxidase. But one of the most elegant studies came out of Dr. Wong Riley's laboratory, where she showed that what she did was she measured the effect of photobiomodulation at different wavelengths. So from 670, 660, 670 to 880 nanometers on cytochrome oxidase activity and ATP content following treatment with the metabolic inhibitor cyanide, a very low dose of cyanide. The cyanide reduced the response. It didn't kill the cell. It reduced the ATP synthesis and the cytochrome oxidase activity by about 20%. So, so that second dot here, I can't actually point it out, but at the 728 mark, that's essentially where, yeah, that's where you're going. Clark, just go right on up to the red dot, the red one. Yep, right there. That's what we saw with cyanide alone. Okay, that tells you that 728 isn't doing much. 670, 880, 830, 850, those are all working quite nicely. So essentially what we saw was that the action spectrum for cytochrome oxidase activity, the action spectrum being that of the activities that happen, and ATP content corresponds with the absorption spectrum for cytochrome oxidase, which says it's absorbing there and something's happening. So that positive relationship between the wavelength efficacy and the absorption spectrum very strongly supports the hypothesis that cytochrome C oxidase plays an important role in the mechanism of photobiomodulation. And there have been many studies beyond that, which I'm not going to go into in the talk. I would certainly address them in questions that also document that. Next slide, please. Now, I told you it gets a little complicated. So if you go to hypoxic conditions, so in disease states, you sort of shift from normoxic early on to hypoxic conditions. And what happens is that cytochrome oxidase can then be a molecular switch in response to oxygen. And so normally under normoxic conditions, It's generating ATP, it's making water, and it's doing that electron transport function. But under hypoxic conditions, this was some very nice work by Dr. Poynton's lab, those conditions really accompany severe disease and pathological states. It switches to a CCO-NO pathway, so it increases NO synthesis. And the presence of this switch explains an increased nitric oxide production after the irradiation of hypoxic tissue with low intensity yellow light. That would be 590 plus or minus 14 nanometers is the peak that they saw. So NO is released from the mitochondria, and NO is a really awesome signaling molecule, very much involved in vasodilation and signaling and inflammation. And so you've got, again, you've got mitochondrial function improved, and then you've also got the nitric oxide anti-inflammatory vasodilatory effect going on as well. Next slide, please. We know that photobiomodulation, so I want to address the fact that a lot of these studies have been done at various, at the far red, and there's an abundance of information at 670 nanometers and 660 nanometers, but there's also a lot of studies that have been done at the near infrared. And this is a study that came out just a year or so ago, looking at mitochondrial function in single cells and actually looking at mitochondrial membrane potential. So again, more support for the idea that the mitochondrial bioenergetic state is improved. And so this shows that photobiomodulation has a direct effect on this membrane potential. It also, not shown here, but it also alters reactive oxygen species. And in these particular studies, vesicle transport in adipose stem cells. But this ability to regulate membrane potential directly within minutes of treating with 830 light indicates that there's a direct action on the mitochondria, there's a direct change, an improvement in membrane potential, and that would be an improvement in ATP as well. Next slide, please. So we know that mitochondrial dysfunction plays a really important role in retinal aging and disease. Mitochondria are sort of the alpha and omega. They run the show. It's not just ATP. They communicate with the nucleus. They regulate cell death. They're very important in signaling and redox processes. We couldn't metabolize amino acids, lipids, and nucleotides without them. And of course, you know, as an extra little goodie, they do energy metabolism. The retina is the highest consuming organ in the body and is totally reliant upon mitochondrial function. So with aging and retinal disease, there's an increase in mitochondrial dysfunction. There's an increase in oxidative damage and corresponding decrements, decreases in the antioxidant and repair systems. And that results in the loss of retinal function, retinal dysfunction. Retinal cell loss then leads to visual impairment and blindness. Next slide. So what I want to do, and I'm going to try to do this quickly, is talk about the importance of mitochondrial dysfunction and photobiomodulation in these various diseases. So my labs looked at diabetic retinopathy, methanol intoxication, AMD. And what you get here is, and glaucoma and hereditary retinal diseases, that would be retinitis pigmentosa, what you get is this vicious cycle. So you get mitochondrial dysfunction leading to oxidative damage, leading to more mitochondrial dysfunction, and eventually you end up at the bottom of this, this cycle, which is in cell death and blindness. Next slide, please. So we started, or I'm going to go over, sorry, I'll step back here. There are a lot of experimental models of human eye disease. Experimental preclinical research is essential for understanding safe and efficient progression into the clinic. There are limitations. Animal models don't have a macula. You can't have age-related macular degeneration because there's no macula or fovea. They have a visual streak, but they do undergo degenerative changes. And there are differences in some of the inflammatory responses. There are lots of animal models that have been developed for research in ophthalmology, retinal injury models, AMD models, retinitis pigmentosa models, diabetic retinopathy models. And photobiomodulation has been investigated in many, if not all, of these models so far. And what we've seen and what other labs have seen is an improvement in a variety of outcome measures following photobiomodulation treatment. The other thing we've seen is no evidence, and I can say for my lab, we've done studies that have lasted 2 years, no evidence of toxicity in terms of retinal dysfunction or histopathology in these animal models. Next slide, please. So the first model we worked with was one that I developed in my lab, and methanol is an incredible model of mitochondrial dysfunction in the retina. Methanol is oxidized to formic acid. Formic acid poisons cytochrome oxidase. Okay, so what could be more perfect than poisoning the molecule that photobiomodulation stimulates? So we had spent years studying photoreceptor mitochondrial disruption and looking at the decrease in retinal responsiveness and the changes in the retina in response to methanol intoxication with the increase in formic acid. So formic acid poisons cytochrome oxidase. It's a mitochondrial poison, and we have a model for it. Next slide, please. So the perfect study would be to say, we got a model for this. There's a light that stimulates cytochrome oxidase activity. We should be able to shine that light on these rats that are going blind from methanol toxicity, and they shouldn't go blind. And I have to tell you, I did this experiment 25 plus years ago, and I was stunned because I really was absolutely blown away. I'd been studying a lot of other compounds to look at, and light now became my drug because 670 nanometer treatment delivered at 5, 25, and 50 hours after they were intoxicated at a low dose, 4 joules per square centimeter. Okay, we put these little lights on, resulted in a significant recovery of retinal function. It went back, in fact, after 24 hours later, this is at 72 hours of intoxication, where if you and I had this much methanol, we'd be blind. The rats are blind generally. They're not. It went back to nearly control levels. More importantly, or equally stunningly, there was no, the damage that is done to the mitochondria in the inner segments in the intoxicated animals and is done to the photoreceptor cells did not occur. The photobiomodulation protected against that. Next slide, please. Then we moved on to an actual disease state in collaboration with colleagues in Australia. And we looked at retinitis pigmentosa. We looked at an animal model of retinitis pigmentosa, the P23H rat. This has a mutation in the rhodopsin gene that's the same as the human mutation. So the closest thing you can get in a rat to a rat-human retinitis pigmentosa model. And in this model, the rod photoreceptors begin to die early in development, and then it slows. So what we did was we treated them during this early period of development, postnatal day 10 to 25. We treated them in this case with near infrared light, the 830 nanometer LED array, about the same, again, the same dose or around the same dose, 4 or 5 joules per square centimeter. And what we did was we said, let's ask about retinal metabolism, retinal function, and retinal histology or morphology at P30. Next slide, please. What we saw was that, and one of the things, again, really strong evidence here in this red, green, and blue slide to the left, that the effect is right on the mitochondrion. So the normal retina is shown, the redox state is shown in blue. And you can see that there's a shift to the left, which means it's reduced, it's not working as well in the retina from the P23H transgenic rat, in the diseased rat, the retinitis pigmentosa animals. And with treatment with near infrared light, that's green, I would have done the colors differently, the shift goes right back to normal retina. So the redox state is adjusted back to the normal response. We were quite stunned by this. And if you look at the response of in the middle here, we're looking at the electroretinogram, so retinal function, and you can see that and the top line is the animals treated with the near infrared light, and the bottom line, and this is the B wave here, is the animals treated, the control animals, the sham animals. And then again, if you go to histology or histopathology, in the P23H rat retina, there are 50% fewer nuclei in the outer nuclear layer. What does that mean? There are 50% fewer living photoreceptor cells, mostly rods, because they're rod-dominated animals. If you look at the animals treated with photobiomodulation, their outer nuclear layer is the same. It's not pictured here, but the numbers of cells in the layers, you could overlap that with the control retina. So the P23H rat is protected against this loss of nuclei just by treating it during those first 10 days, basically, of development. Next slide, please. We've also looked at age-related macular degeneration, and we're going to hear a lot more about this. It's the leading cause of death in the RP and photoreceptors. It's increasing astronomically, and there are no approved treatments for early to intermediate forms of dry AMD. Next slide, please. What we did for these studies was we used a knockout mouse model that had a transcription factor that's really important in the antioxidant process. So if you knock out the NERF gene, what happens is in the eye, you get an AMD-like pathology. The RPE degenerates, the ERG goes down, and you get drusen-like deposits. So we took these animals, you have to age them. We took them at 10 months, treated them with red 670 nanometer photobiomodulation daily for 3 months, and the sham animals weren't exposed to light. And then at 13 months, so 10 months, 13 months, we actually measured the ERG, and the PBM significantly improved retinal function as measured by that response. And those improvements were documented in both photoreceptor, that's the A wave, and bipolar and Mueller glial cell function, that's the B wave. Next slide, please. There's been some studies actually that came out of University College London looking at another model of AMD. This is a knockout, a mouse model that is a knockout for complement factor H. And what you see is that 670 nanometer light reduces the outer retinal inflammation. So there's a tremendous, and it's easiest to see actually looking at the pretty picture here, showing that with the light, that it's just not inflamed. It's not lighted up at all. The C3 expression was significantly reduced in Bruch's membrane and the photoreceptor outer segments. Next slide, please. The other one is a study that actually came out of a laboratory, a very productive laboratory in Brazil here, and showing that, and a really very elegant study showing that photobiomodulation reduces vascularization in the chicken chorioallantoic membrane. This is a very nice model of a vascular network, and you can just use these, just shine it right over as it shows in the illustration. The chorioallantoic membrane in the egg, and the vascular network can be analyzed. This study also has a very nice positive and negative control. The negative control was treated with phosphate buffered saline. Positive control was treated with Avastin, basically. Good way to reduce the angiogenic effects here. And what you see is that 2, 4, and 8 applications of 670 nanometer PBM at 5.4 joules per square centimeter resulted in a progressive decrease in the vascular network, similar to the actions of Avastin. And so you see a very nice reduction in new vessel formation. This is very exciting implications in terms of neovascular diseases, including AMD and diabetic retinopathy. Next slide, please. And then there's some work by Gu et al. That shows in another model, this is a toxin, sodium iodate model of induced retinal damage. And what he shows is that multi-wavelength. So we've shown 6, 7 red, near infrared. Now you mix red and near infrared together and you get protective effects as well. He looked at the outer nuclear layer, ganglion cell layer, and essentially what he showed was he poisons it with sodium iodate. He kills off the photoreceptor cells. He actually kills off the ganglion cells as well. And animals that were treated with photobiomodulation, the multi-wavelength photobiomodulation, protected against the loss of those cells. So it inhibited cell death. Next slide, please. And he went on really to show that it reduced retinal apoptosis. So it decreased the number of cells dying after that. It decreased the loss of rod bipolar cells in the internuclear layer, if you look across here. It inhibited photoreceptor degeneration. So it was protecting. So the big picture was the slide before, the actual layers of the retina. Here you've got the nice stains that are showing you that individual cells are protected with the multi-wavelength PBM. Next slide, please. And the last thing that they showed was that PBM doesn't damage these cultured RPE cells. And they looked at doses in terms of cytotoxicity from 1.1 joules per square centimeter to 18. And they essentially showed equivalent proliferation and no evidence of cytotoxicity. So at the cellular level, you're not seeing any damage. I think we're on to you, Clark. It's your slide now.
Thank you, Janis. That was a very nice summary of a lot of different animal studies by various groups around the world. I'm going to pick up and move more towards the requirements and the clinical data that we've been generating at Lumithera. So at Lumithera, we developed the Valeda light delivery system. It was considered a Class 2A device in Europe based on the safety assessments of the device itself. That Class 2A device was tested both for ANSI 780.36 and IEC 6471 for light exposure safety. It is not a laser, it is LEDs, and it meets the eye-safe requirements based on all industry requirements. It doesn't provide, as photobiomodulation is stimulating cellular outcome measures, but it does not provide a thermal treatment or produce local cellular damage, as we've seen in the animal studies as well as in our clinical studies for up to several years. We obtained a CE mark in Europe. The review was granted after our technical file, which included 3 clinical trials, the Torpor 1 and 2, which use the same wavelengths in the LIGHTSITE I study, as well as relevant literature. And it was reviewed by a UK practicing ophthalmologist. ANVISA also reviewed the technical documents subsequently and granted a registration for our ability to treat and sell in Brazil. The review basically takes into consideration the safety, both preclinical and clinical trials that are available in the published literature, as well as what the manufacturer has created. The FDA follows the same type of review process. In our case, the device was not classified. We actually submitted to the FDA a PMA in September of last year. The FDA, after their initial review, came back and reduced the class of device to a Class 2 device, which basically means that it's unified around the world as a global classification as a Class 2 device. The FDA then requested us to submit a what they call de novo application. And a de novo application is for novel products to come onto the market with a defined or definitive benefit to risk profile. It also allows you to put in special controls to protect the device. Those special controls include labeling, non-clinical benchtop testing, as well as clinical performance. Now, difference between a device and a drug, and I think in several cases, when you look at the data, this does resemble a treatment similar to what we see in drugs. However, in the device area, there is more effort in the safety and the process that go into the control of the device. Typically, a pivotal clinical trial is required for U.S. approval in the FDA's mind, and it differs from the drugs where you would require 2 phase 3 randomized clinical trials. Right now, we have completed our pivotal clinical trial. It is not approved yet for photobiomodulation for dry AMD. It is in front of the FDA. The de novo application has been submitted. They're under review, and we anticipate the review to be complete and approved in the fourth quarter of 2024. Now, we also, as a manufacturer, we do not dictate the price set forth by the clinicians to treat patients. We have settled a price for the capital item purchase of the Valeda. We also have a treatment credit fee for each eye. We sell those through our customer store at a price for the US dollars. It's $261. That includes a full round of 9 treatment sessions for both eyes. The cost is basically about $14.50 per eye, or $29 for both eyes. We do not impact the physician's own practice and business. They can set their own price for the service of providing the Valeda treatments. We recognize that that can vary from country to country, from clinic to clinic, but I would say by and large, we've now had approximately a little over 21,000 sets of 9 sessions treated over the course of the last 4 to 5 years. And most of the doctors charge what they would normally charge for an office visit to receive and provide a treatment with a trained physician or technician. What does the Valeda do? It provides a little over 4 minute and 10-second treatment that is broken up into 4 phases. We did create a multiple wavelength device. So the first 35 seconds is pulsed with 590 as well as the eyes are open, and then phase 2 is closed, and it's a continuous 90-second treatment. The total treatment for those 4 sections are 4 minutes and 10 seconds. Both eyes can be treated one after the other in less than 10 minutes. The multiple wavelength approach, we've already talked about some of the clinical and preclinical data that supports the different wavelengths. Each wavelength has a different unique spectrum in terms of tissue scatter and tissue absorbance. We created a multiple wavelength approach because we think each one of the 3 wavelengths has unique and distinct attributes. 590, we've heard already and seen data about its role on NO synthesis, as well as its potential to act as a second messenger to increase blood flow or local vasodilation. The 660 is tended to be approved and targets the oxygen binding site. We've seen that with the profile over scanning the wavelengths from about 500 to 1,000, where you see a peak at 660. And 850 is a near-infrared peak, which really helps drive electron transfer at copper site A of cytochrome C oxidase. So both of those wavelengths do approach cytochrome C oxidase, but they approach 2 different phases, similar to any other protein, where it may have 2 different substrates. We want to make electrons active, and we want to be able to improve the affinity for oxygen binding to help increase the proton gradient that drives ATP synthase. So we believe it's a very directed approach for 3 wavelengths that all has distinct properties that can be beneficial in the overall clinical outcomes. So what I want to focus on now is the LIGHTSITE III. It's our pivotal U.S. clinical trial. It was conducted in 10 centers across the United States, all retinal doctors that are involved in research. Many of them are your colleagues that you know from many of the international meetings. The study was FDA approved after a review of our IDE. It's a double mask, randomized prospective study. It's got a sham control arm, multi-center, and the study was designed to look at both safety and efficacy of photobiomodulation in intermediate patients with dry AMD for up to 2 years. The trial design basically had the patients come in for 6 rounds of treatment every 4 months. They did 9 sessions over the course of 3 to 5 weeks. The Valeda device was built so we could actually do a randomized clinical trial. So it actually, in the clinical mode, it has a sham, and the sham is reducing the yellow by about 10x and 100x for the red, and the near infrared is turned off. And the reason those are reduced by that level is they still are bright, they still have some activity, but the patient cannot determine between one or the other, which is the lower fluence wavelength. The study particularly looked at intermediate dry AMD patients. So these patients typically had to have some vision loss, 2032 to 2100. They also could not have central involving GA. We wanted tissue that was viable. These were patients that had basically had been diagnosed about 5 years earlier. And we did look at a modified Ferris simplified risk analysis. Most of these patients had moderate to high risk for progression, with a risk factor of 3 to 4, considering the treatment eye and the fellow eye as well. The patients were enrolled 2 to 1. They were randomized 2 to 1 because these patients are losing vision, and we felt that this was an appropriate design that minimized the risk for those sham patients that would lose vision over the course of being in a trial for 2 years. They went through 4 rounds of treatment, and then at month 13, we did an interim analysis. The study was continued for 2 more rounds, and we'll now talk about the final 24-month, which was 3 months after the last treatment round. So basically, most of the patients were Caucasian, female over male by about 65% to 35% overall. The age typically is around 75 in our clinical trials. This is a third of the LIGHTSITE trials, and all of them have been around that 75 year of age as the mean age. In this study in the US, AREDS vitamins are quite prominent, and so we did stratify. So each group had similar amounts of either AREDS supplements or no supplements. To be in the study, they had to be stabilized either 3 months in AREDS supplements, or if they were not on it, they stayed off of them for the 2 years. The BCVA starting letter score was around 70 letters. And what we see is after the first round of treatment, we're starting to see an increase in best corrected visual acuity. The letter score and change from baseline about 3 letters. By the second round, we've seen this before, we tend to get increasing effects after the second round. And by about the third round, we're stabilizing. This is the modified intent to treat group. So this is the most conservative, taking all comers that at least had gone through 1 round and came in for 1 post-treatment 1 outcome measure. What we did see is a couple letters in the sham. And while we know that this is an active control, and we've seen that before, we'll talk a little bit about this as we go forward in the slides. But what we tended to see is about 10 months, both in the European and this trial, we start to see a further separation between the 2 groups. And in this study, we saw the sustained benefit out to 24 months with the PBM treated group, where we started to see a diminution and a further separation at 24 months. So while we had a significant benefit at 13 months, pre-specified p-value was 0.02. You can see at month 24, the p-value was even stronger at 0.0015. And this is, of course, at 24 months, this is 3 months after the last round of treatment. Now, what's interesting is as we looked at this, as in the earlier studies, we had decided to look at the starting vision and how PBM may impact that. What we see in this study is that we had patients that were 61 to 70 or greater than 70, 70 to 80 letters. And typically when we look at these patients that have really good vision, we have a ceiling effect in which we cannot look at a very large increase in BCVA because they're already starting out very healthy. In fact, what we see is about a 4 to 5 letter increase, but you also see that the sham treatment, this is where we particularly saw that early sham benefit. It tends to dissipate, and by 24 months, you can see it is going away as the patients are progressing and the treatments have stopped. Interestingly, if you look at the 61 to 70 letters where you remove the ceiling effect, they're starting with a little worse vision. We see now about an 8-letter gain And in the sham group, we only see about a 1-letter change. And in fact, it mirrors kind of the natural progression that we know in AMD, dry AMD patients in the intermediate stage. And that was maintained, that 7 to 8-letter increase or delta, net delta, was maintained out to 24 months. So it's also very important where the patients are starting. You can start earlier where you can see some vision loss, but you're not going to see a 3-letter gain or a 3-line gain in those early patients. But as you move forward, you can see some very nice and prominent maintenance of a significant increase in PBM, induced BCVA. We looked then at a Cox proportional hazard model post-op and looked at the survival rates. And what we saw was that the hazard ratio showed about a 53% reduction in the onset of vision loss. So not only were we seeing vision gain in these patients, the sham group tended to show a more significant vision loss over the course of 2 years. So at 13 months, we saw about 7% had lost greater than 5 letters, but by 21 and 24 months, we're up to 15 and 18% of those patients had lost significant vision. In addition to that, we looked at a variety of anatomical. All the centers were using the Heidelberg Spectralis, and using Duke as our independent imaging center, they actually qualified the patients. They were masked to all of the imaging. They used the Heidelberg and FAF as well as fundus photograph to determine new onset GA. And at 13 months, we were seeing about 1% of the PBM patients were progressing to new GA. 10% of the sham patients were, but by 24 months, we were now up to 24% of those patients had progressed to new GA. Only 6 to 7% in the PBM treated group had progressed. And interestingly, all of these patients with the Ferris modified scoring system had a score of 4 or maximum high risk for progression to disease. In the sham, it was, again, predominantly high risk, but we did have some 2 and 3s that did go ahead and progress to GA. Lastly, if we look at the hazard analysis for new onset GA, there's about a 73% reduction in risk to moving to new GA over 24 months using the Valeda light delivery system. So in summary, when we look at this, we see a nice sustained or maintenance of vision improvement of about 5 letters out to 24 months. It's 5.9 letters at the end of 24 months. But if we look at the distribution in the modified intent to treat, about 60% of the patients had about a 5-letter gain, 18.7% had a 10-letter gain, 5.5% had a 15-letter gain. So we mentioned more patients lost vision in the sham at month 13 and on out to the end of the study. We saw a reduction in the patients progressing to new GA. We also saw a reduction in iRORA develop over the course of 2 years, a reduction in the small number of patients that had iRORA that converted to CRORA over the course of 2 years. So the biomarkers were also in favor of PBM, and we saw a very favorable safety profile. The safety profile is shown on the next slide. We saw a few very minor and mild AEs in the ocular area. One of them that I want to point to is the conversion to neovascular. We did see in the sham eyes 3.6% of the patients, 7.5% in the PBM, and 8.3% in the non-study eyes that we followed, 52 non-study eyes out for the course of the study. What I do want to bring out is the difference here between the PBM conversion to wet and those in the sham eyes. And so when we started this study, we did not stratify by the fellow eye being wet. In other words, those patients that were higher risk for conversion to neovascular. And what we saw was that there were a total of 16 eyes randomized into the study. 12 of those were randomized into the PBM, only 4 in the sham. So that's a 3x or 3-fold higher ratio in the PBM to sham group in terms of high-risk eyes. When we actually normalized and adjusted that to the sham for the same risk ratio, the amount of expected conversion was 7.3 versus 7.5. We now are continuing to look at those patients that have gone forward in our LIGHTSITE III study, which is an extension for these patients to continue for a third year. And I can tell you that all of the sham, PBM, or non-study eyes are falling in the same line. Sham and PBM are around 10 to 11% conversion, and the non-study eyes is around 13%, all within the 10 to 15% that you would see in normal population. So we can't say that we're reducing, but we are trending a little bit lower than the non-study eye. Here's the rest of the AEs that were ocular. Again, very minimal, very minor, similar in both the sham, PBM, and the non-study eye group. So what about safety? In addition to our trials, there's been 2 randomized clinical trials in AMD that's enrolled a significant number of patients. COVE enrolled 66 patients, and 346 were enrolled by Ivandec. Both of those had a small component of neovascular, and what they saw in both of those studies were reductions in edema and bleeding in eyes that were diagnosed with neovascular AMD. There were no safety concerns in either study. So while we can't say that we're reducing neovascular, there is some preclinical data as well as a couple studies that are suggesting there may be some benefits in neovascular conversion as well. Those studies also showed that in reductions in edema and restoration of tissue architect was seen in a DME study. In Central Cirrus, recent case study came out showing benefits there. And we've seen a variety of preclinical data from Janis already this morning on the potential that this treatment could be used in other indications. Currently, we're involved in a variety of other studies. We're supporting a study from Dr. Uthan in Oslo. They're looking at a long-term randomized clinical trial on the conversion to neovascularization. So it's enrolling right now 121 patients. I think they've got about 30 or 40 patients enrolled already, and is going to be looking at treatments every 6 months for over 2 years with 1-year follow-up. We also are seeing some other long-term studies on safety of photobiomodulation. One of the areas that we can point to very nicely is in the use of PBM as a first-line treatment against radiation and oral mucosis. Studies there have followed PBM now for over 15 years without showing any development of toxicity or AEs or development of secondary malignancy. So there is a lot of safety data around photobiomodulation, not only in the ophthalmology space, but in other walks of life. And then finally, then as we look at trying to do these trials, we designed the Valeda light delivery system so we could mask. It is difficult to mask a light-based study because if we can see the wavelengths, they're probably also hitting some of the targets we're after. We have seen recently that some lower dose studies have indicated benefits in ocular vision on a short 2-week treatment, which indicates that our sham control is probably an active control that may have some early benefits and then wanes as the patients progress. What we have done is, I think, a very good randomized clinical trial. These are results showing other anatomical benefits. This is from the LIGHTSITE I study showing nice reductions in drusen. We can see deposits in drusen reduce, particularly if it's late stage and there's a lot of drusen. In the earlier patients with less drusen, we see numerical changes, but we did not see in the 13-month study a statistical significant reduction in drusen overall, about a 3-fold reduction, a 4-fold at month 24. This paper recently came out showing drusen reductions and demonstrating from a group in Paris, Dr. Sued's group, showed nice reductions over a course of about 6 months in reductions in drusens in specific patients. So we do believe that the benefits are making the tissue healthier, improving energy, reducing the amount of drusen that is spit out and deposited extracellularly, and hence creating a more healthy retinal tissue. Lastly, we continue to build a robust dossier of data. We now have 5 clinical trials that we have completed. We're continuing to add to that. Each trial we learn a little bit from. The current trials have been supported by other publications using the Valeda light delivery system in 2023, and we're seeing more that are coming out in 2024. We have extended the LIGHTSITE III for another year. It's an open label study with a crossover for the sham patients. And we have initiated a registry study in Europe. We're expanding that into South America and the US, in which we're collecting data from 500 to 1,000 patients, not only retrospectively, but prospectively to look at things such as lesion, GA, lesion growth size, conversion, and progression of disease. So I think in summary, what I'd like to say was we're continuing to build the data that supports the use of the Valeda light delivery system in photobiomodulation, ocular disease, and disorders. Right now, we are providing something that, based on studies in the UK, we can reduce healthcare costs, we can improve the quality of life, and I think in conclusion, that provides us with an opportunity to continue to grow PBM's use in ophthalmology conditions, as well as expansion into non-ocular indications. We're seeing that globally. PBM so far has been shown to be safe and effective in multiple preclinical and clinical studies. We think it provides a safe and effective treatment for intermediate patients, and PBM may represent a broader platform for other acute and chronic inflammatory indications that will need to be further determined in future clinical trials. So with that, and I know that was a lot, but I appreciate everybody's attendance, and we're here to answer any questions as well. Thank you.
Perfect, Dr. Clark, Professor Janis, thank you for all the content exhibit. We mostly had 3 questions from the Dr. Rubens Belfort, who's a very well-known professor here in Brazil. It's a very important doctor that is now using Valeda. He's asking the first one, I believe it is already answered, but if you would like to comment, he's asking if any animal model with using the same wavelengths with Valeda.
So one of the things that we've seen is that the different wavelengths have been used in multiple animal models. All 3 wavelengths have not been used in a animal model, but individually they have been shown to have benefits in either rodent animal models. The problem with some of the animal models, as Janis said, is that while as a pharmacologist, toxicologist, you try to choose the best model, the rodent models do not have the fovea. There is no one model for AMD. Different groups tend to, in the research, use the red light because it's safer. If you're using near infrared, you have to really protect your lab to make sure that any of the beams don't bounce off a mirror or window or something like that. I have been involved in a previous life in near infrared, and we do know that that is very safe. It went through a 1,000-patient stroke trial in the US and Europe. So I've been involved with all 3 wavelengths in different aspects. And for us, we can utilize data that's been generated by a variety of researchers. We don't have to reinvent the wheel every time.
Great. Another question for Dr. Rubens is, any information of Leber optic neuropathy considered as, any information Leber optic neuropathy considered as mitochondria?
Let me address that. So Dr. Belfort, it's good to talk to you again after coming for the LHON Brazil group in early 2000 and working with Solange and Adriana. We did a brief trial there. We looked at the effect on 6 individuals who were affected with labors, were blinded with labors. So we had complete approval for the trial, and but there wasn't a huge risk because we were looking at individuals who already had lost vision. However, there's some peripheral vision. At the end of a week's worth of treatments, we did get subjective reports of improved vision. One person said that he could actually see the colors and stripes of his granddaughter's dress, and brought that information up, which he couldn't have just been informed of. And some of our tests were positive. Mostly they were not definitive, and there was some concern about the neuron-specific enolase, which is a biomarker of neuron damage, and there was a bit of a spike in it. So Alfredo Sadun and I discussed this and decided that before moving forward with anything with labors in humans, we wanted, now that there is actually an animal model, we wanted to actually test it in an animal model, or at least test it in cells, because I wouldn't want to make anything worse with someone who had a disease that could tip one way or the other.
Perfect, perfect. I believe it was already commented also in the presentation, but he also asks if there is any potential indication for anterior segment.
Yeah, I think, Janis, you want to talk about that, and then I can add to it?
Well, I guess if we're going to sort of divide things up, there's a lot of work going on now there's some work going on in terms of vision and myopia. There's a lot of interest in corneal abrasions. So, I mean, that's really taking the anterior segment way out to the outside. But the first place that photobiomodulation showed remarkable success was wound healing. And essentially, corneal abrasions and corneal disruptions are just that, wound healing. And so I think that's an exciting area. I think there's a number of other areas. I'm going to let Clark sort of jump in. I live happily in the posterior segment. I'm gradually struggling to move forward in the eye.
Yeah, I would just add to Janis is that, we've been focused on obviously AMD. We think there are very other diseases or injury to the eye, particularly that are addressing inflammatory pathways that would be very amenable for PBM. Obviously, there's a lot of work going on in myopia over in the Pacific Rim. But I think what Janet indicated is spot on. Our two founders, besides myself and two other engineers, we had two ophthalmologists. One was an anterior segment surgeon, and his first use of photobiomodulation was in PRK. And he showed he could reduce recovery or reepithelialization from about 4 to 5 days to about 2 to 3 days, and reduce inflammation. And so that was really where he was interested in photobiomodulation, because it does inhibit collagenase. So when you look at the TORPA 1 and 2 studies, the 3 wavelengths we chose and the fluences are based on products that were taking out of a skeletal muscle and a dermal application that individually had been approved and shown to be safe. So we felt that all 3 wavelengths addressed a different target that was particularly important in degenerative chronic diseases. But I do believe wound healing is a very interesting opportunity for patients. We have videos of a Bengal tiger, very valuable aged Bengal tiger in the LA Zoo that had a corneal ulcer and it was treated. And we do know that there's some other opportunities, including cancer. It's big in protecting against radiation damage. So protecting against radiation pellets that are used in ocular cancer, while a small indication could be very useful as well. So I think there are other applications that will develop over time. We strongly believe it's a platform. It's not going to work in everything, but done with the right clinical trials, it may have multiple uses in ophthalmology.
Great, great. We received another question from Dr. Abenur, who's a very important name also. Other than patients with new AMD, neovascular AMD in the other eye, what patients are in particular risk of developing neovascular AMD in the treatment eye? I believe he's asking about the risks the patient that has only AMD, dry AMD, develop the neovascular form.
Yeah, so again, we chose the 590 because of its ability to be involved in stimulating NO. But in addition to that, there's data out there that shows that it inhibits VEGF expression naturally, which would give it a separate mechanism similar to the anti-VEGF that could be important in reducing the amount of neovascularization in a dry AMD patient. So we do believe that we've got some components in here in the preclinical data that Janis has shown as well supports that it may have some benefit. Now, we chose to go after dry AMD because, quite frankly, anti-VEGFs are very powerful, very good drugs. We felt that the dry AMD patients didn't have many options, and so this is something that we could bring to the table. There are interesting comments coming out of researchers that those eyes that are stabilized, that you may not have gotten back all the vision you want, the Valeda may improve that. So I think now that we're showing that we're slowing the underlying progression to GA, one of the things we want to consider is how important is this as a additive to anti-VEGF to maybe augment its benefits, to maybe modify the sequence or need for repeat injections as frequently in those patients that you can't stabilize. So I think there are some interesting discussions that need to be said about Valeda's potential use in wet AMD as well, that will spur some additional research.
Yes, in addition to what Dr. Clark told, I believe the question that everybody would like the answer, and I believe unfortunately it won't be answered because how many patients we are treating with Valeda nowadays, and they are not converting due to the treatment. I believe that's one good one to calculate, but it will never have a proper answer because we will never know if the patient were treated.
William, what I can tell you is we just had a discussion yesterday in front of about the same number, about 50 UK doctors. And one of our speakers was Dr. Andrew Luff, who was the first ever Valeda purchaser. And he's treated now over 200 patients. And it isn't a clinical study, but he's gone back and he's only had 2 patients convert to neovascular. He's only had 3 patients that he's had to stop because of conversion to geographic atrophy. So his data was very supportive of the low risk of conversion. The study that we're doing in Oslo, Norway, should help. That's a very specific randomized clinical trial. But that center has done over 350 patients, and they have enrolled now 100 of them into the registry study. So we are collecting data from use from centers that have high volumes to be able to look at not only retrospectively, but prospectively conversion rates, progression of GA, lesion growth size, really to expand the data to provide a ongoing foundation of further data to support its use.
That's great to hear because with that kind of statistic calculation, we would have the number of the patients we removed from the line of using anti-VEGF due to the conversion to the dry AMD to wet AMD.
And I'll be honest, I'm a pharmacologist by training. Anti-VEGF is a very powerful, very good drug. And but there are patients who don't respond to it. There's patients that are difficult that you can't do the maintenance, and so the mechanisms are different. We're looking at an underlying mitochondrial approach. So you could argue that while you can use anti-VEGF to stabilize that wet eye, the disease is still marching forward. So I think what we need to look at, and ultimately what you're going to see, it's going to be a multitude of whether it's vitamins, pharmaceuticals, devices such as the Valeda that are going to have to be used because the disease itself is very complicated. We have both genetics, we have age, we have comorbidities. There's no one magic bullet here. And what we're trying to do is provide an opportunity for patients that we know what the etiology is going to be to come in and maybe give them back some vision and slow down the progression of disease.
I'm going to step in for just a second because I've always felt that one of the, as a pharmacologist whose drug is now light, but I still think about drugs, is one of the advantages of photobiomodulation is that it can be used in concert with other medications. You don't have to worry about drug-drug interactions. You can add, a light treatment to a drug treatment. And I think that's a very powerful model to move forward with.
Yeah, I would just add to that is because of the age of the population, they're going to be on multiple medications, and so adding anything can add another level of complexity.
Right.
Perfect. The questions that we received for Q&A were those that I told. I believe that's it. If you guys would like to add anything over.
Well, I think I would just like to thank Janis. She's been one of the pioneers in photobiomodulation. 20 years ago, working in methanol toxicity, she asked an important question. I can prove if light works, and I think there's been a variety of photobiomodulation experts that we have talked to and worked with. We are very excited about what we can do. We want to continue to build the basic scientific and clinical data to support it. We're very happy to have the opportunity today to speak to some of our Brazilian colleagues and welcome the opportunity anytime to come back and speak again. And finally, thank you for taking your time this evening to be with us.
I'd like to thank you too, and thank Clark. I'd like to thank my Brazilian colleagues because actually the very first introduction to photobiomodulation that I had in terms of a conference was in Brazil, where photobiomodulation had been used in dentistry for years. And so I was speaking, everyone believed everything I had to say about the benefits of light. The other thing is the first time I did some of these studies and realized that it was going to work in a rat, I said to myself 20 years ago, I want to see this in a clinic before I ever think about retiring from this field. And so I want to thank Clark for putting it in the clinic. I really appreciate that.
I would like to thank you guys, Professor Janis, Professor Clark, and all the audience. Thank you, everybody, to reserve a little bit of time to be with us. That was very clarifying webinar and very clarifying session. I believe it will be very good for our audience because some of the doubts that were said in few days ago can now be relieved. Thank you all.
I think we'll be more than happy to make the slide deck available, as well as I believe that for those that would like to attend that couldn't, this will be recorded, so we'll be able to provide this for anybody that's interested in listening.
Right.
Perfect. We will help on spreading the word.
Thank you
very much.
Thank you very much, Professor Janis, Professor Clark. Good to see you all.
All right. Thank you.